Good afternoon, everyone. I am Almilin Madani, and for today, I am going to discuss mobility, distribution, and fate. So, for today's lesson, our objectives are first, identify factors related to environmental toxicity, second, differentiate bioaccumulation and biomagnification, and third is discuss persistence and environmental interaction. In ecosystems, toxic waste have caused substantial damage to animal and plant populations. Such waste overwhelm natural restorative processes, destroy habitats, and reduce populations of sensitive species outright or inhibit their reproductive success. And some common examples of toxic waste are mercury, lead, and hazardous or toxic air pollutants. So now we are going to tackle the factors in environmental toxicity. So in the environment, the concentration, transport, transformation, and fate of a chemical are affected by several factors. And factors which affect overall toxicity may be described under these four headings. First, factors related to chemical. Second, factors related to exposure. Third, factors related to organism. And fourth is the factors related to environment. So first, we will now discuss the factors related to chemical. The physical chemical properties of a compound that are considered important include its molecular structure, solubility, ionization, and vapor pressure. Other properties of chemicals, such as rate constant for hydrolysis, photolysis, evaporation, absorption, and partition coefficients, are also important and affect environmental concentration of toxicants. Actually, all these characteristics affect the toxicological and pharmacological properties of the compound. And the nature of chemical reaction is determined by the functional groups of the substance, and its toxicity is a response of organisms consequent upon reaction between the compound and certain part of the organism. So next is the concentration or dose of chemical. A toxic substance produces its toxic effects after its interaction with appropriate receptors of the organism. The effect of directly dependent on the concentration of the chemical at the target site, and concentration at the target site is related to the amount or the dose of the chemical administered. In environmental exposures, an estimate of the dose can be made from the measurement of environmental and food concentrations as a functions of time, and it also involves the assessment of food intake, rate of inhalation, and the deposition and retention factors. So now, let us move on to selective toxicity. Selective toxicity results from biological diversity and variations in the response of living cells and tissues to different chemicals. There appear to be involved two important mechanisms for the selective action of a chemical. These are presence or absence of specific receptor or target sites and the factors that may be responsible for the distribution and change in the concentration of the chemical at specific sites. This results from processes such as selective absorption, translocation, biotransformation, and excretion. So now let us move on to chemical interaction. In nature, sometimes organisms are exposed not only to one chemical, but to two or three chemicals at the same time. And these chemicals may interact with each other, and such chemical interactions may have great toxicological significance. For example, effect of ethanol and carbon tetrachloride on liver, or effect of cigarette smoking and asbestos exposure in the lung tissue, or the effect of malathion on EPN on when given simultaneously to test animals, are far more toxic than the sum of their toxicities when given separately. Now, let's move on to factors related to exposure. Any toxic substance cannot show its effect unless it comes in direct contact of the organism having a natural environment or maintained in the laboratory for bioassay. This contact reaction between the organism and chemical is called exposure. In toxicity studies, the most significant factors related to exposure are: first, the concentration of the chemical; second, the kind, duration, and frequency of exposure; and last is the exposure routes. So, having contact with chemical, biological, or physical substances found in air, water, food, or soil that may have a harmful effect on a person's health. Examples of environmental exposures include UV radiation from the sun and cigarette smoke, radon, or other forms of air pollution. So, the factors related to organism. Toxicity of a chemical is generally evaluated against a particular organism or a group of organisms. The toxicity of a chemical varies according to the size, life history stage, age, sex, and health and nutritional status of the organism. Acclimation of the organisms to laboratory conditions prior to bioassay also plays an important role. The same species or organisms collected from different habitats may exhibit variations in their limits of tolerance to the chemicals. Certain individuals of a species may be susceptible to a chemical, while others may be resistant. Generally, the most desirable species for toxicity testing are those that have relatively short life cycles, mature quickly, thrive in laboratory aquaria, and those that can be easily handled and are small as adults. The most important considerations in the selection of test organisms for bioassays are: first, their economic, ecological, and recreational importance; second, their sensitivity to the material or environmental factors and their consideration; third, their geographical distribution, abundance, and availability throughout the year; fourth, knowledge of their environmental requirements and culture methods for rearing the laboratory; fifth, their general physical conditions and freedom from disease; and last is their suitability for bioassay test. Mm. So let us now proceed to the factors related to environment. There are many abiotic and biotic factors of the environment that may modify the toxicity of chemicals, and these factors are: first, the water temperature. The water temperature is expected to greatly affect the toxicity of cyanobiotics. The increased water temperature increases the solubility of many substances, and it affects the chemical form of some and govern the amounts of dissolved oxygen in water. So our next factor is dissolved oxygen. We all know that oxygen is essentially required for respiration by organisms, right? So, any physiological change affecting the rate of respiratory flow of aquatic organisms may affect the concentration of the toxicant at the gill surface and thus the toxicity of the chemical. So, third is the pH. A rise of one pH unit actually within the usual middle ranges increases the proportion of ammonia or NH3 by about six folds. Hence, six folds increase in the toxicity of substance. So the fourth factor is the salinity. The greatest differences in chemical characteristics of freshwater and seawater may be expected to enormously affect the toxicity of chemicals. But in general, salinity does not really significantly affect the toxicity of xenobiotics. However, a particular species may be more affected with changes in water salinity. So next factor is the hardness of water or water hardness. The total hardness of water has little effect on the toxicity of most of the chemicals except for metals. And the toxicity of ammonia, phenols, surfactants, and pesticides has been reported to be affected the least by the hardness of water. So the last factor is suspended and dissolved substances. So, toxicity of metals is often greatly decreased by the suspended and dissolved matters present in water because of absorption and binding, while other xenobiotics are much less affected. So, now let us proceed to the difference of bioaccumulation and biomagnification. So bioaccumulation takes place in a single organisms over the span of its life, resulting in a higher concentration in older individuals, while biomagnification takes place as chemicals transfer rather from lower trophic levels to higher trophic levels within a food web, resulting in a higher concentration in apex predators. So, bioaccumulation refers to the buildup of a toxic chemical in the body of a living organism, while biomagnification is the increase of the concentration of a toxic chemical when going along a food chain. So, example of bioaccumulation and biomagnification is this situation. So, mercury is a harmful chemical present in the water. Bacteria feed on mercury, and this is bioaccumulation at this point. Then, small fish feed on bacteria, which increases the amount of mercury the small fish have. This now becomes biomagnification. So, next is let's talk about persistence. Persistence in terms of environmental protection refers to the length of time a contaminant remains in the environment. Contaminants can pose significant health threats towards people, animals, and the environment, so their fate, transport, and persistence is of particular interest. Persistent bioaccumulative toxic substances or PBTs are chemicals that break down slowly in the environment, accumulate in humans and other species, and are toxic. Although only a limited number of PBTs are currently receiving attention. Humans, domestic animals, and wildlife are more likely to be exposed to a chemical if it does not easily degrade or is dispersed widely in the environment. The structural characteristics that enable a chemical to persist in the environment can also help it to resist metabolic breakdown in people or wildlife. Now let's move on to interactions. In discussions of the environment, the synergistic interplay between problems receives little attention, even though it could prove to be the most important factor of all. Synergism occurs when two or more processes interact so that the product of their effect is greater than the sum of their separate effects. A biota's tolerance of one stress that tends to be lower when other stresses are present. For instance, when low sunlight reduces a plant's photosynthetic activity, the plant becomes more prone to the effects of cold weather. And cold weather also increases a plant's vulnerability to low sunlight. We know all too little about environmental synergisms, but also the ecologists cannot even identify many of the natural manifestations, let alone document the impact. If we can discern possible synergisms in the environmental heave us ahead, we shall be better able to anticipate and even prevent some of their adverse repercussions. So that's all for my topic. These are my references. I hope that you have learned something from my lesson for today. Thank you and God bless.